So welcome to video 3 in our course G06 on RNA Seq transcriptomics. So now we're going to consider RNA Seq experimental design, and we know that there's a lot of sources of variation in these kind of experiments. We have to consider the RNA selection and depletion strategies, the relevance of replicates, especially biological replicates, batch effects if we do different sequencing runs at different days. If we're using stranded or unstranded libraries, typically it's stranded. Read types that we're using, the read lengths that we're using, the depth that we have, and the need for more careful QC with RNA data rather than genomic data because of the tendency for that to degrade more quickly. So an illustration of kind of steps that we might expect in our RNA-seq library prep are outlined here, where we extract first our total RNA and then enrich it for mRNA using either poly-A enrichment steps here, which is the more common one, although the option is RNA depletion or poly-A depletion as well. Then after this step, we can go onto library construction and sequencing, and there's differences between whether we're using, say, an Illumina type short read library or maybe a longer nanopore library. So the different prep kits, depending which library approach is being taken. We cannot analyze RNA data using either approach, but there's slightly different approaches to generate these reads here that are of interest. So what we can consider there is that most of the batch effects that we get from RNA sequencing are associated with the generation of these libraries that we get. So we have to consider that during QC later. So that can be associated with RNA extraction, the ribosomal RNA depletion, poly-A enrichment, conversion to cDNA, amplification processes as well. And therefore, if we alter the steps, the kits, the buffers, and so on, we get these batch effects and technical artifacts. And that applies to broader sequencing experiments, especially ones that are maybe you're comparing data from two different studies. Really needs careful thought. And especially when you consider if we're looking at different poly-A or ribosomal RNA depletion approaches, the levels of ribosomal RNA are very, very large. So our section of segments that contain mRNA is typically smaller overall, and that's often our RNA that's most of interest. But there might be experiments where you're looking at other types of RNA as well. So for poly-A enrichment, remember that we're dealing with mRNA binding beads contained a T, which is kind of shown down here. This is our oligo bead with the T's here, and then the A's attached onto there. The transcripts, the ribosomal RNA, etc., transcripts without a poly-A tail are washed off. At the per right, we typically use one of these kits, New England Biolabs Next Ultra RNA kit, Next Ultra Directional RNA kit, or the Illumina TruSeq Stranded RNA kit. And this is a major preference, and ribosomal RNA may also be possible, but is not essential if this is done already. At our institute as well, we're looking at using stranded libraries, but note that they differ from unstranded ones, because here for the stranded ones, we know whether the reads are from the sense strand or the antisense strand. So we know that their direction of these reads is from 3' to 5', whereas these ones are going from 5' to 3'. For unstranded libraries, we don't know that direction, and that creates kind of some uncertainty over which whether the genes are aligning to the sense direction or the antisense direction of our reference genome or transcriptome. And that can occur, and in rare cases, we do know that there are overlapping genes, and therefore, if we have overlapping exons, that can mean that reads might be misassigned in terms of determining their origin. And these are called cisnatural antisense transcripts, NATs, as well. We also know from different studies that gene expression values from in this example, from one representative cell in 10 studies where you have normal expression values, shows that the extent to which there's different genes being expressed varies across a lot across different studies. So the minimum threshold is given by the red line in each study, which is empirically determined for each one. That's kind of our management detection threshold. You see for some, like this one here, you can see that the level of expression is much lower, whereas for this one over here, it's much larger. So that means we have to consider how we compare between different studies, different samples, and different batches. One way to achieve this, in simple terms, is to add in biological replicates. So if we have two replicates, our estimate of the variance will be a certain width. But if we have more replicates, like 5 replicates, we'll find that that variance decreases. We have a better estimate of the true mean for our particular sample. So as we increase the replicates, we get a better understanding of our particular, say, cell line or our host response that's of interest. Achieving this can sometimes not be as clear as you might wish. So for example, if you're looking at this diagram here, where you have an epinorth with some sort of aliquot, and we send that onto 2 different dishes where maybe things are grown further and they're taken out onto different slides, we consider how many biological replicates are actually achieved in this experiment. So at the end, we've got 6 samples down here, and how many of these are biological replicates or technical replicates or biologically different samples. We can also do the same thing here, where we have 3 different tubes at the top, then we have pairs of aliquots all the way through, and then the analysis of these 6 down at the end here. So how many biological replicates are present and how many technical replicates are present as well. And then how can we increase the number of biological replicates here? We know for RNA-seq that technical replicates are not useful. That one RNA-seq one gives a decent, should not be massively skewed by technical artifacts in general. Technical replicates, remember that we have the same mice and we take the same sample from the same mice and put that same sample in 3 different tubes. Biological replicates means that we have the same isogenic mouse line exposed to the same pathogen or whatever, but our samples are taken from 3 different mice. The biological variation is going to be much larger, and it's better to sequence more samples in general than, say, just biological replicates, as just depending on your context. So generally, having more biological replicates or samples with low coverage is better than having fewer with higher coverage. You can see that here. If you have a number of reads, your capacity to detect differentially expressed genes, something to this false discovery rate, FDR, increases a little bit, but the number of replicates increases it more. So jumping from 2 to 3 to 4, 5, 6, 7 replicates increases our power much more effectively. So, of course, when we think about power for RNA-seq, because we're dealing with many different genes, power testing for RNA-seq is not necessarily straightforward, and it's unique to different genes and transcripts. And of course, we have this problem of estimating the variance for each gene and transcript again as well. So this is non-trivial. What we can do is look at the relative association between the model counts versus the variance for each isoform. Where at one level we might expect no association between the count and the variance, but in reality, you may get the variance may increase with the number of counts that we get. And we have to always pause here and just think about our original question, that if we had infinite sample sizes, in principle, all genes and isoforms would have some level of altered expression if we're comparing 2 different groups. So we have to always have to think about what are we actually trying to test here. Are we trying to identify the genes that change most in response to, say, exposure to a particular pathogen, or what precisely? You know, if you had infinite sample sizes, what would a particular approach actually tell you in terms of your biological question? To assist with this, there is one useful calculator here. As there's a shiny version of it. There's also one you can implement in RStudio. But in this instance, if we're dealing with 10 subjects, where we're looking for testing for differential expression between different groups, where the minimum average read counts for the genes of interest is 25, the dispersion is set to 0.5, the ratio of geometric normalization factors is 1. The number of genes here is set to about 20,000, which is typical for many host genomes. We've only got 100 genes that are actually prognostic, that are informative here. And we set our minimal fold change to be 2. And then our power for this will actually be about 0 if we have a false discovery rate of 0.05. So 10 samples for a typical sequencing run where there's limited read coverage will have no power at all. If we increase that to 30 with the same context, that will increase to 0.4. So remember, the power is our capacity to detect true positives. So if this will mean we only get 40% of the true positives. It doesn't really tell us here about our false positives. We can know that the rate is 0.05, but it depends on how many we have that, how many genes we're testing overall. And if it's 20,000, we can therefore realistically expect that 1 in 20 of them would actually appear as false positives, which could be a lot. If we increase it from 30 to 50, our power jumps to 0.85, so we're improving there. And we can graphically display this in this manner. So it's the same parameters here. Well, the sample size is shown as 47, but it's roughly the same, that once we have maybe 15 or so samples, we start to get some power to detect effects. And we see here, if we've 50 at the top, we really jump up higher. If we alter our criteria from this fold change of 2, if we look at a fold change of 2 and our coverage increase, our read coverage to be 100, minimum of 100. So if we have more reads, our power is, if we have 15 again, is still very low. It's down here, but a power here, if we have 38 or so, is much bigger. It's about 0.8. If we increase our criteria for our fold change of at least 4 for those 100 genes, we find that our power jumps up much more. And now our power kind of surpasses this point of 80% at around 11 or 12. So you can see that the kind of context of how differentially expressed genes, how the extent to which there should be differential expression present, which we may not know in advance, that affects our power. So the estimating our power is non-trivial. One thing we can be sure about is that if we're trying to estimate some data and we have 2 replicates, if we increase that to 4 replicates or 2 replicates shown by the blue line here, this is a green line, and this is the read count. So if we had our value 25 here before, you can see that our power for 2 replicates is here around measurement uncertainty, I should say, is about 0.25. Increase it to 2 replicates, it falls to maybe from 0.26 to 0.23. But if we increase that to 4 replicates, we see that falls considerably to 0.18. If we increase the read depth between 2 replicates, the green and red, with the same read depth, we see that we get this big jump here, this big fall. And whereas if we have double the read depth for our 2 samples there in blue, we only get a small fall. So increasing read depth does not really help our power much. We need more biological replicates to tell us more. And that might be, those biological replicates might be cell lines that we're trying to, a single cell line we're trying to examine, or it might be a different context or some sort of epidemiological experiment, but more samples is better. So then to look at really at gene level differential expression, the ENCODE experiments suggest that we need at least 50 million paired reads per sample, and these would be stranded. If we have at least 3 biological replicates, then we can get away with 7.5 million paired reads. Obviously now all our reads will be much longer than 50 base pairs, and more biological replicates will help where possible. To detect isoform level differential expression, which is more refined than gene level, we need about 30 million paired reads per sample for known isoforms and 60 million paired reads for novel isoforms. So you can see getting novel isoforms at a level, it's going to be quite tricky. Linked to this is our experimental design, and one thing that's always important is that if we have different groups, and different sequencing platforms or different approaches, different library preparation approaches, we should be down here and mix those samples with different preparation approaches or different contexts together in different sequencing batches. are possible. If we link the different groups to different sequencing approaches and runs, they'll be confounded. So for the above one, if we consider this, we might have differences between the different groups, but they are correlated with the different batches. So we can't discriminate between batch effects versus true group effects. They're conflated with each other. Down here, if we look at the first box, so if we see these differences where the differences between the blue, green, orange, and purple groups are marginal and sensible, we can say that, well, it's possible to correct for the batch effects between different ones. That maybe the initial group had a low level of cDNA available and there was more in group 2, and intermediate in group 3, we can kind of correct for that. However, we may find artifacts where there's some sort of other batch effect in, say, the origins of group 2 and group 1 and group 3 here. So that can also happen too. So we kind of have to consider the experimental design and to, per possible, share different samples from different origins and preparation approaches across different batches. There's other studies that have looked at this. So this is an example of a study down here where they looked at PCA analysis of single-cell RNA-seq data and a cluster by sequencing experiment, sequencing instrument, where this blue clump is high-seq, and this other red one, it's a different sequencing approach. And that's linked to the detection rate. That does, the detection rate kind of increases a bit for Illumina, and for the other one, it increases differently. And you can see there's clear difference between these different groups. And that sort of artifact means that it kind of ruins the whole approach of looking at transcriptome analysis in this context, because you don't know to what extent the different sequencing devices have kind of obscured what's really going on. So one consideration on this is to pool samples where possible. So if we take our samples through a pipeline here, we have our total RNA, we fragment them to deplete the ribosomal RNA, reverse transcribe to get cDNA, build our index libraries, and we have them all separate. We can create more technical variation here when we pull the samples at the very end. If per possible, we can take our RNA, fragment it, pull the index the samples together, and do these steps together for the same samples. That will reduce the technical variation associated with our experiment, because we'll reduce the differences between different samples that are caused by the differential treatment here. And unfortunately, none of our pipetting techniques are perfect. We always will make slight differences in terms of the volume that's given to different tubes, and those collectively will lead to differences that are not real in our terms of analyzing our output at the end. So that kind of concludes our video on RNA-seq experimental design. Please see our next video for more on counting and normalizing RNA-seq data.